Good morning, class. Thank you very much for listening for this video. So, per today's discussion, I'm going to talk about DNA, RNA, and proteins. So, first na pumunta tayo sa ating DNA. So, DNA stands for deoxyribonucleic acid, which is, we got here a deoxyribose, which is actually the sugar content or the sugar base of a DNA, and nucleic is the main base of a DNA. So deoxyribonucleic acid. So it is actually the blueprint for life, which is commonly found in our nucleus together with the chromosomes. Okay. So as you can see here in the cell and the nucleus, andyan po si chromosomes natin. And then of course those strands sa chromosomes, we got the DNA. So of course, so meron po tayong gene na rin as our unit for our heredity. So there you go. So so the structure of DNA is actually a polymer of what we call as a nucleotides. So meaning, so nucleotides po natin is the base or the basic structure of our DNA. So ito po yung tsura ng nucleotides, class, na. So this is the structure of a DNA. We were going to observe it one by one. So first, we got the basic nucleotide structure, this one. So as you know, this class, we got a nitrogen base, which is this side. Ayan. A phosphate, and as well as our 5-carbon sugar. Since we are dealing with a DNA, so we call the sugar that is connected to our basic nucleotide structure as a deoxyribose. Okay? Deoxyribose. That is the sugar found in our basic nucleotide structure. So, itong nucleotide structure sa class, magdidikit-dikit po siya to form a polymer. And of course, this polymer or this long strand of polymers is what we call na as our DNA. Okay pa? So, each nucleotide, once again, composed of nitrogen base, phosphate, and 5-carbon sugar. So, now, we're going to discuss naman yung ating nitrogenous base. As I told you earlier, since si ating nucleotide has nitrogen base, we got our 4 bases, okay? First is the purine bases, which is actually our adenine and guanine, while our pyrimidines are the cytosine and thymine, okay? So, tandaan po natin po yung ating mga bases po na yan. So, pag sabing purine, adenine and guanine, pag sabing pyrimidine, cytosine and thymine. So, for example, sa isang nucleotide base, meron po tayong phosphate, at meron po tayong adenine sa taas, at andito po si deoxyribo. So, dikit-dikit po yan, tatlong yan. Okay po? Now, we have what we call as our complementary base pairing class. So, when it comes to complementary base pairing class, si adenine po, lagi po siyang naka-pair po kay thymine. Ayan. While si cytosine po, lagi po siya naka-pair po kay guanine. So, ang ginagawa ko niya na mnemonics is at CG. So, ganun po siya para matandaan niyo po na. So, at CG. So, si adenine, partner po si thymine. Si cytosine, partner po si guanine. So, at CG. Malinaw pa. So, ayan. There you go. So, si at and CG. So we got here, as you know, this class, no, we got our nitrogenous base, our deoxyribose nucleus, and our phosphate molecule. Ayan. Now, as you notice, no, ating DNA structure is actually a double helix structure, which is once again discovered by Watson and Crick. So, but then again, we got an issue way back before between Watson, Crick, and of course our Rosalind Franklin, diba? Yung ating photo 51. So, but then again, itong discovery ng DNA structure was actually, of course, ang pinangalan po na discover ay si Watson and Crick. So, ganun po siya, na. Next, there you go. Now, let's go towards our DNA replication. So, during our DNA replication class, this actually happens when there is actually a need to replicate or duplicate a new cell. Di ba nga, tandaan natin sa ating mitosis, syempre, and meiosis as well, di ba nagkakaroon tayo ng separation or division of the cell. So, actually, even 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 si DNA and of course the RNA na pag-discussan later is of course mag-replicate. So, as you notice class, in DNA replication, so the very first enzyme that is actually happening is it reads the coil or the helix is actually what we call as our DNA topoimerase. So, this DNA topoimerase class will untangle the coil. Kasi nga po, tandaan natin, ang tsura po kasi ng ating DNA is actually a double helix. So, just double helix na gantong normal na tuloy-tuloy lang class is actually being twisted po. Paikot po siya. So, ito pong enzyme na ito, purpose niya po is to untangle the coil. So, DNA topoimerase. So, once the DNA was, of course, untangled na po doon galing sa coil niya, the next enzyme that we call as helicase unzips this DNA. So, etong helicase po na to, ang gagawin niya po is, of course, hihiwalayin niya po sa gitna yung dalawang yung ating double helix na DNA. Mamaya may picture dyan ng process kung paano po nangyari yun, na. So, ayan. Thus, of course, from double-stranded na DNA, ngayon, pag nahiwalay na po siya ng ating enzyme class, of course, magiging dalawang separate na po siya na single-strand na DNA. Okay po? Ayan. Of course, then, of course, i-prepare na po nung ating bagong replicated na dito tawag nating DNA replicated na po. So, dun sa ating base pairs. So, once again, so if, kunyari itong side nito, no? If si ating adenine is nandito, si guanine, si thymine, yung mga ganyang class, no? Ayan. So, once na mahiwalay na po ni helicase si ating double-stranded na of course, na DNA. So, mag-replicate ngayon po siya. However, it's actually not just simple replicate class. Of course, there's actually a need for them to be paired dun sa ating mga pairs and of course, bases. So, once again, si adenine, partner kay thymine. So, si G, kay C, A, G, and another C. That is actually how replication works. Okay? Ayan. After na mag-replicate po siya at magkaroon ng bonding po, next is si DNA ligase na po is actually follow behind to bond the nucleotide together. Meaning, kung si ating helicase po yung nag-unzip, naghihiwalay, si ligase po yung nagkakabit po. Maliwanag po ba doon? So, the process of DNA replication is fast and accurate. An error occurs in only about 1 of the billion nucleotides. Okay? So, it's because of what we call as DNA polymerase. So, si DNA polymerase po, lagi niyo po double check if ever na nagkaroon po ng error pagdating po sa DNA replication. Okay? So, maliwanag po tayo doon. So, ulitin natin. So, at the first stage, meron tayong DNA polymerase to untangle the coil. The next stage, we got the helicase, so an enzyme to unzip or to separate this double-stranded helix. So when this double-stranded helix has been separated, of course, it will now replicate itself. So when it has been replicated, of course, it will be replicated based on their base pairs. So, if it is an adenine, it will be paired into thymine. If it is guanine, it will be paired in cytosine. So, once lahat po siya na paired na po, of course, DNA polymerase will follow through. So, si DNA polymerase mag-check ng mga mistake. If ever nagkaroon po sa maling pairing po during the replication. Okay? And lastly, DNA ligase will follow through as well at the last, of course, to bond the nucleotides together. Maliban na po ba tayo doon? So, DNA replication begins at specific sites called the origins of replication. The copying proceeds outwards in both directions, creating replication bubbles class. So, in the parent DNA strands open up as daughter strands grow on both sides on each bubble. So, meaning lang po niyang class, the replications actually working as this one. Ang nangyayari po is that baliktaran po yung nangyayari. No, si new strand po ng ating rather ng ating base pair, si new strand na to is going outwards, while of course going inward naman po si ating old strand po. No, so siya po ay separate o opposing yung kanilang direction. Okay? Yan. DNA replication occur before the cell divides. So, once again, si DNA replication po, ayan, nangyari po siya before the cell division, ensuring that the cells of multicellular organisms carry all the same genetic information. So, the resulting double-stranded DNA molecules are identical. It's always identical class, okay? Proofreading and error checking mechanism exists. So, such as, for example, si ating DNA polymerase class. Next is si ating RNA structure. So, kung tinatawag nating deoxyribonucleic acid si DNA, we call RNA is our ribonucleic acid. With that said, ang ating sugar po kay ating RNA instead of deoxyribose kay DNA, si RNA po ay we call them as ribose. Okay, it is actually quite similar to DNA. However, there is actually different nitrogenous base in an RNA. And aside from that, single strand po si RNA compare kay DNA na double stranded po na. So, ito po yan, class. Ayan. So, ribose po, instead of deoxyribose, Dex is, of course, the presence of the uracil. So, as you notice, class, wala po tayong thymine sa ating RNA. So, instead of using thymine, sa RNA, we use uracil. Thus, our pairing would be GC. So, gano'n, tandaan ninyo na. So, mamaya kasi magkaroon tayo ng transcription. Okay? So, ayan. Yan po si RNA class. So, RNA is single-stranded and much shorter than the DNA. So, ayan. Diretso na po tayo kay central dogma. So, pag po si DNA, nag-process po into another DNA, we call them as replication. From DNA to RNA, we call them as transcription. Ayun pa, tuturuan ko yun yan sa next lesson kung paano mag-transcript. Now, next is lastly, RNA to proteins, we call them as translation. So, we got actually 2 amino acids or 2 proteins, 20 proteins rather, na ipapakita ko sa inyo kung ano yung mga yun na. Yan. So, the 3 different RNA molecules involved in protein synthesis, ayan, are mRNA or the messenger RNA. We got the rRNA or the ribosomal RNA. Actually, is actually not that quite impactful, pero isama na natin siya. And lastly, of course, si tRNA or si transfer RNA, which is actually responsible for translating. Okay pa? So, the first step of the central dogma class of the protein synthesis is, of course, the transcription. This occurs in the nucleus. The transcription po, once again, it will transcribe from DNA to RNA. While the second step would be the translation would always occur in the cytoplasm. Since the translation, dito na po na-involve si ribosome. Kasi po from RNA to protein na po siya. Tandaan natin, the site for protein synthesis is ribosome. Okay pa? Dumunta sa ating transcription. So, transcription always occurs in a nucleus. So, the first step is DNA unwind section. And, of course, the mRNA will be formed to the base pairing. So, paano po siya, class? Ay! Nakuha naman. Ganito po siya, class, na. Okay. So, assuming na magkakaroon tayo ng ating for a while. Assuming na meron po tayong dito na isang strand, ng DNA at meron po tayong mga bases na A, D, C, G, A, C. But this is a DNA, no? In transcription class, diba, in translation class, diba nga, ang ginagawa natin, pinipair natin siya doon sa kanyang base pair. So once again, si adenine will be paired with thymine. Thymine will be paired by adenine. So cytosine will be paired by guanine, while si guanine will pair cytosine, and so on and so forth, class. Now, this is a DNA replication. However, in DNA, in, of course, transcription rather, tulatan natin si ating A, T, C, G, A, C, si DNA. We will transcript from DNA to mRNA, class. Na, yan. 1, 2, 3, 4, 5, 6. Now, so, as you notice, class, if we will pair this adenine, diba? Normally, pag nireplicate natin siya, it will be paired in thymine. However, in RNA, we don't have thymine. Thus, we will use uracil instead. We will use U. Yan ang papasok si U. Okay? So, si thymine magiging pong A. So, may guanine pa rin po. May cytosine. Of course, wala po tayong thymine sa ating mRNA. So, we will use U. And lastly, our guanine. So, this is the transcription. I hope maliwanag po yan. Now, you can do some practice online to how to transcript. So, let's continue. So, once mRNA is formed, enzyme in the nucleus remove the introns, the non-coding message, and leave the exons, which is the good message, class. Okay? Ito po siya. Now, next we go to our genetic code. So, each 3 consecutive base of mRNA is a code word codon. So, paano po si codon, class? So, sa example natin kanina, sa previous slide, we got the ATCGAC, TCGAC. Ayan. Kunyari ito yung ating, genetic code, class. The codon, class, is every 3. So, we call this 3 group, itong triad na to as codon. This one as well as codon. So, the genetic code consists of 64 codon. So, sobrang dami nun, no? So, mahirap siyang, sobrang dami niya pag transcript siya at nireplicate. So, yan po. So, but only 61 code can be coded into amino acids. So, 3 codon acts as signal to stop the process. So, ayan. 1 codon, AUG, codes to for methionine, which is also, of course, the start signal. Okay po? Mamaya, papakita ko yung table about this one. Yung table ng ating codons, class, no? Ito yun, class pala. Ayan. I will provide this one. No worries. I will provide this one on our exam. This table. Para sa start and stop codon. Okay? But this actually fixed when si AUG at si stop codon natin. Now, next is, of course, our translation. So, tapos natin kay transcription. So, I hope na mali na po sa atin. No? So, next natin is translation, class. So, translation actually from mRNA to protein. So, translation, synthesizing a code from amino acid according to the sequence of the nucleotides in mRNA. So, we will translate rather the mRNA, always mRNA po, into protein. Now, this always occur po sa ating cytoplasm ng cell. And of course, it will involve yung ating ribosome. Since of course, si ribosome po is the process for protein, synthesis. Okay? So, ganito po siya, class. Ayan. So, si ribosome po is the site for protein synthesis. So, ito po si ribosome po yan. Well, this one is our mRNA. So, kakatapos lang po niya galing ng ating transcription. So, dadaan ngayon po siya kay ating ribosome. So, itong si ating ribosome class, ngayon, pag dumaan si mRNA dyan, may lalabas po na polypeptide, yung ating peptides na mga bond ng amino, bagong protein na. Okay, isa-isahin tanyang class, no worries. So, we got what we call the transfer RNA. Si transfer RNA class actually transport molecule which holds specific amino acids or it holds specific proteins class. So, 20, we got 20 amino acids class, okay? So, I will show you this one for a while. Ayan. So, these are the 20 amino acids na, of course, dinadala ni transport RNA, ni tRNA. So, madami-dami po siya. Si alanine up until kay tyrosine class. So, yan. So, no worries, you don't need to sauluhin tong mula short hanggang van der Waals volume. Just the name itself lang kailangan, no? So, next is, of course, this codon. So, each codon represents po ang isang specific na of course stop or start na na command, and of course it will always represent po ang isang amino acid. Now, this transfer molecule or tRNA, siya po yung nagdadala po, no? Siya lang po yung nagdadala ng class para bigay po doon kay bagong strand ng, ayan, ito po yung class. Meron po yan dito. For a while. Paano ba ito? Ayan, there you go. So, si mRNA na to class, galing pa po yan from transcription. Transcription, once again, from DNA to mRNA. So, once dumana po siya dito, on the way on this one, ganito po ang kanyang direction. Itong si ribosome naman po, papunta naman po sa opposite direction. Ganon po palagi ang kanyang ang kanilang galaw. Lagi po silang opposing ang galaw nila. So, we got the free tRNA napupunta po dito kay ating ribosome. Once again, si ribosome po ang ating, sorry for that one, ang ating place or the site for protein synthesis. So, once yung tRNA po ay nakapasok na po kay ribosome, i-pair niya po ni tRNA, kung sinong tamang pair po nila. Ay, may pusa. Wait lang. Sorry. So, i-pair niya po ngayon itong pair na to, itong tamang pair na yung class kay kay mRNA. So, si tRNA po, meron po siyang specific na pairing o base pairs. For example, as you notice on this one, sorry. How to do this? There you go. So, as you notice on this one class, no? So, si ating adenine was paired by uracil, and of course, and so on and so forth. No? But, then again, you will always base kung sino pong itong ating amino acid based po doon sa code na nakalagay po kay mRNA. Hindi po kay sa code po ni tRNA. Uulitin ko, ang naka-assign po na amino acid ay binabase po kung ano po yung code na nakalagay kay mRNA, hindi po kay tRNA class. So, once si methionine kasi class is start codon. So, ito po yan. Is AUG, which is actually start. Start codon po siya. That's why andyan po siya na. Malinaw po ba? So, instead of using UAG, hindi po pwedeng gamitin yan. Once again, kay mRNA codon lang po tayo pwede magbasa. So, once maibigay na po si amino acid, once na ma-pair na po yung mga bases po dito, at syempre dala-dala ng tRNA yan, of course, bubuo na po ng bagong chain or peptides. Tatawag nating peptides ng, of course, amino acid, forming the new protein class. So, I hope malinaw po yun. No? So, isa-isahin natin from the top. So, of translation. So, mRNA leaves the nucleus to migrates to ribosome. So, of course, si mRNA na po is na-pair na po siya from transcription. mRNA binds to small ribosomal subunit, which is of course the ribosome. Technically, tRNA brings an amino acid to ribosome where anticodon on the tRNA binds to the codon of mRNA. So we call actually this one, itong daladalang tRNA, as anticodon. Anticodon. Okay? Hindi yan nabasihan ng name ng ating protein. So, next step, the amino acid bonds to its adjoining amino acid to form growing polypeptide molecule. And P of the tRNA without the amino acid is released from the ribosomal. Of course, pag nagkaroon na po ng van der Waals connection between yung molecules ng amino acid nila, magkakabit-kabit na po siya hanggang sa, of course, aalis na po si tRNA from its line. Of course, wala na po siyang dalang amino acid. So, lastly, other tRNAs bring ito amino acid to ribosome to complete the protein molecule. So, that technically said na, of course, other tRNA, papasok na po siya dun sa ating ribosomal subunit. Okay? So, ito po yung protein translation. Ayan. So, from here, ayan, no? Ayan. Up until, of course. So, we got a free tRNA. Meron tayong anticodon. Meron tayong mRNA, which is ating codon. So, i-pair niya po yan. Magka-pairing po sa kung yung pair po nila, na. So, once na mapunta po sa tamang pairing po nila, itong si ribosome, mag-go against the flow. Kung dito ang flow ni ating mRNA, ang flow ni ribosome papunta po against, na. So, once si ribosome is, of course, habang dumating si ribosome, may dumating nga tRNA. Si tRNA, kakabit po siya sa kanyang anticodon, and of course, iiwanan yung ating yung ating amino acid. So, once na naiwan si amino acid dito, of course, lalabas ngayon dito yung bagong chain ng, of course, protein polypeptides. So, I hope na marina po ito na. So, protein synthesis will always occur in the cytoplasm of the cell. Okay, so this is the summary of the protein synthesis. Ayan. So, we got here the cytoplasm. Ito yung ating nucleus class na. So, mRNA transcribed to DNA. And of course, si mRNA po, dadaan po ngayon dito, class. Doon ang direction po niya. Sorry for that one. Next, si ribosome po, opposite direction po galaw ni ribosome, class. So, once again, pag dumadaan si ribosome, class, meron po tayo nagprepare na tRNA. And so on and so forth. Up until magkaroon tayo ng complete na bagong polypeptide chain. So, that is the protein synthesis, class. Now, I hope na malinaw po ito, class, kung paano po gawin, of course, magsaulo ng ating central dogma. No worries, on the next lesson, I will discuss to you further for that one, okay, in face-to-face. So, I hope that you will review well on this certain topics. Medyo challenging siya. So, thank you very much for listening. Have a great day ahead.